present the Award of Excellence. This award, obviously, one of the highlights of awards from our society. And this year, the award goes to Dr. Dennis Newbold. He's going to give us a great presentation on his work, obviously, on spiraling, as we're all familiar with Dennis' work. Dennis is a research scientist emeritus at the Stroud Water Research Center in Avondale, Pennsylvania. He studied at Swarthmore College, where he earned a bachelor's degree in engineering, where he first encountered the concept of a riparian forest buffer. Then at Cornell, where he earned his master's in hydrology, he modeled oxygen dynamics in the Cayuga Lake, and finally moved on to Berkeley, where he earned a PhD in aquatic ecology, where he found that a 30-meter riparian forest protected macroinvertebrate diversity. Dennis did his postdoc work at Oak Ridge National Laboratory, where he introduced him to Jack Webster, and then his new concept of stream nutrient spiraling, which led to a series of both field and theoretical investigations of spiraling. Since moving to the Stroud Center in 1983, Dennis has studied the spiraling of fine particulate and dissolved organic carbon, and his work has extended to models of mayfly seasonal synchrony, nutrient dynamics in Costa Rica, and studying how riparian forest buffers intercept nutrients and sediments and preserve stream geomorphology, habitat, and ecosystem processes. So here to tell us about spiraling at distance travel, I am more than happy and ecstatic to introduce Dr. Dennis Newbold. Dennis, take it away.
Thank you, Checo, for that great introduction. I'm really honored, of course, to receive this award, and it came as a big surprise. So I want to thank those who nominated me and helped with the selection. And I want to thank the Society for Freshwater Science for being what's really an exceptional organization from the standpoint of collegiality and mutual support. It's always been a great group of people to work with. And I also want to thank the Society for its role in this recent crisis with Black lives. I think we're all aware of the damage that racism does in our society. Not the Freshwater Sciences Society, I mean in our country and in the world, and support efforts to remediate that. But we're now in a time when it's not enough just to be on the right side of the issues. We really have to step up and do our part. And I applaud the society for taking those needed steps. So in processing this award, I realized that it's not an award about me, it's about spiraling. So what I'm going to talk about is spiraling, but of course, you're going to get it from my perspective. So why don't we go to the slides? Okay, before I even start, I want to mention that what you see in this picture, this is in Idaho. You see that man in the blue in the background is Bert Cushing. And sadly, Bert passed away back in September. But he features in my talk today, as does Wayne Minchell, who also very sadly passed away in April. So those are two people that were big in my work, and so I'm dedicating this talk to Bert and to Wayne. Well, spiraling fell into my lap. I arrived at Oak Ridge in 1977 to study for a postdoc with Jerry Elwood and to work with Bob O'Neill. And both of them were really excited about the idea that they just learned about that came from Jack Webster, who was at what's now Virginia Tech at the time as an assistant professor. So I got involved in trying to bring this idea into focus so that we could submit a proposal and do some work on it. Thinking about nutrient cycling, this was a difficult idea to get one's head around. Cycling in a stream and transport all go mixed together. And in those days, you might remember that nutrient cycling, some of you might remember, I should say, anyway, in those days, you could have a conversation that went something like this. Do you think it's worthwhile to study nutrient cycling in streams? Well, I'm not sure. Do you realize that anything that's going on a square meter of stream bed is overwhelmed by, it's only amounts to maybe 1/10 of 1% of all the water flowing over top. So I'm not sure if anything's going on there and whether it's really worth looking into. So there was, we really had very little idea about what nutrient cycling in a stream was all about. So to help do that, then we arranged to bring Jack on as a collaborator, and also to come and talk to us at Oak Ridge about this concept that was his. And I have to figure out if I can advance this slide. There we go. And there's Jack. So he came over to Oak Ridge, and we hashed out the ideas. And at one point, he went up to the blackboard and he drew these spirals on the blackboard. And he said, well, nutrient cycling in stream isn't really cycling because it's not closed. By the time it cycles, it's gone downstream. So we really should be drawing these as spirals. And then he said, well, what's happening here is that the ecosystem, the biota in the ecosystem are taking up these nutrients, using them, delaying their downstream transport, and regenerating them to the water. And all this is taking nutrients that would otherwise just be whisking on downstream. So it's retaining these nutrients and using them, and using them efficiently with this relatively tight spiraling. And if the ecosystem is seriously disturbed, then these spirals get looser, and the nutrients would just slide on downstream without being used. And then behind this was Bruce Wallace was kind of in the background. He wasn't there, but he had been working with Jack. And this is probably the first drawing that was published of the concept of spiraling. And what you see here is that they were working with filter feeders. The idea, and we were all benthic ecologists except for perhaps Bob O'Neill. So we all had in mind this notion that of caddisflies as filter feeders, catching a particle, ingesting it, processing part of it, digesting that particle, and having that go further downstream, but perhaps caught by another caddisfly downstream. So that's sort of the idea that's being pictured here in this drawing. So it was really thinking about the benthos and the food web that was doing this spiraling that we were starting out with. But back to these spirals, then I got to looking at these, and a few days later, after Jack had put these on the board, I got to wondering and thinking, maybe we should be able to measure the distance between those loops. And a few days later, with a little more work or a lot more work, I realized that for a nice, well-behaved nutrient like phosphorus that's conservative, doesn't have an exchange with the atmosphere, that we could actually estimate that distance as the ratio of the downstream flux of the nutrient to the uptake flux, that uptake flux being that U on the bottom, or the uptake flux per unit meter, with that W being the stream width. So then the more uptake there would be, then the shorter that distance S, and that actually is the average distance that a particle would travel as it makes one loop of, that is, uptake by primary uptake compartments, going through the food web and then back to a state of dissolved inorganic state where it's ready to be taken back up again. So as that uptake increases, the spiraling length shortens and the spiral gets tighter. So this spiraling length then provides an index of the nutrient use and retention against the downstream transport. So with that idea, we went out to Walker Branch, which is a small stream on the Oak Ridge Reservation, where we had a site-wide license to use radioisotopes. And Jerry Elwood had previous experiences putting P32, the radioactive isotope of phosphorus, into the stream. And here he is injecting a half-hour pulse of P32 labeled orthophosphate to the stream. And we intensively then sampled that pulse as it moved downstream, as I'm sure many people who are listening to this have done since then. And then for the next 6 weeks after that, we intensively sampled the benthos. Here is Bob O'Neill sampling the epilithon, or Aufwuchs, as we called it in those days. And here's a sampling of the fine particulate organic matter. Notice that we're all bundled up in protective clothing. P32 is dangerous, and we had to take safety precautions. So when that slug passed downstream, we could see it disappearing as it got farther downstream and integrating under the curve for these 3 stations and for the other stations. We could see that P32 disappeared from the water column exponentially, which on a semi-log scale plots as a straight line with the slope of K sub W. And the inverse of that slope then is the average distance that a phosphorus atom would travel before being taken up on the stream bed. And we call that distance the uptake length. And in this case, that uptake length was 167 meters. But as I mentioned, our real initial interest and focus was on the whole food web, on the ecosystem as a whole, and how spiraling worked with that. So we had this model of the flow through the ecosystem, through the food web. And behind that, we had the mathematical model. And I won't detail this, but the top equations are for the moving compartments, the water and 2 seston compartments moving downstream. Those are advection dispersion equations. And then the benthic compartments were modeled, the relatively simple linear donor controlled transfer model. And those, but now this model is, so these are first order transfers. And in fact, this is actually, as ecosystem models goes, this is a fairly simple model. And it is simple because what we're actually modeling, we're not trying with this model to model the mechanisms of the ecosystem. We're trying to model only the path, the progress of the tracer through the ecosystem. So the real mechanism being modeled here is the mixing of the tracer as it follows the path of the natural phosphorus, which we take as a given. So we're not trying to explain why the transfers are what they are. We're only trying to estimate what they are. So that makes for a far simpler model than you get with a truly mechanistic model that's actually trying to explain the controls on what happens in an ecosystem. Like, for example, this one, which models simultaneously the carbon, nitrogen, and phosphorus, the stoichiometry of 3 elements, and undertakes questions like, how does the leaf decompose based on the stoichiometry of that leaf, plus the availability of the nutrient in the water. So you get a model full of nonlinearities and much more complicated. This model is primarily Jack Webster's, with some help from Lawrence Lynn and me. But the main point is that the model we actually used in Walker Branch for that food web was really far simpler. Now back to that slug passed downstream, then those curves you see were fit by the model. And then that same model with the same coefficients then fit the data that we saw in the primary uptake compartments after that. So what you see is then the initial label on the left declining over time as the phosphorus left these compartments and was taken up by the consumer compartments. This is in the upstream reach. And at the lower end of the reach, we saw something more like this. What you see is, of course, that the compartments didn't label up as much at the bottom because less P32 got there, but the loss curve is flatter. We flattened the curve, so to speak. And the reason it is that not only were those compartments losing phosphorus, but at the same time, they were gaining the isotope that was being released from upstream. And we saw that both in the data and in the model. The model was able to reproduce that. And that difference from upstream to downstream gave us confidence that we were actually seeing the spiraling process. Now, the bottom 2 panels here are for the first of the primary uptake or consumer compartments. This is the snail, which in those days we called Goniabasis. And of course, it was gaining phosphorus as it from the primary uptake compartments. And then we have more consumer compartments, the shredder collectors. But what I really want to point out here is the filter feeder, the caddisfly diplectrona, which was the only compartment that got more strongly labeled downstream than upstream. And that was an interesting and satisfying result, because what it meant was that those upstream caddisflies were actually feeding on suspended particles that were coming in from above the reach where we labeled it. So they were actually feeding on relatively unlabeled particles, whereas the downstream caddisflies were feeding on particles suspended from within the reach that had a strong label on them. Unfortunately, the diplectrona were not didn't have a high population, so these caddisflies weren't really playing a very big role in the spiraling of the system. And then we see the predators down there at the bottom, which were accumulating the P32, of course, more slowly than their prey. So those curves then were described by this matrix of transfer coefficients that basically describe how fast the nutrient moves from one compartment to another. And then on the diagonals, those are just the transfer or the turnover rates within each compartment. And the reason I show this really is to point out that we estimated all these transfers simultaneously in a single model, which is not the typical way this is done. A lot of food web studies basically estimated on a stepwise basis, compartment to compartment. And this approach that does it simultaneously has certain advantages. One being that once you have a completed model, you can run that model to steady state using not the isotope, but the real phosphorus, and then get the actual estimates of fluxes between all the compartments, as well as estimates of the active pool sizes. Now, as I said, this isn't a typical way it's done. So I was really happy to see that just this year, this paper by Andres Lopez and his colleagues came out, which described the food webs of two Trinidadian streams based on N15 stable isotope of nitrogen tracer work. And that was in some ways much more sophisticated than what we did, because it used Monte Carlo, a Markov chain Monte Carlo approach to estimating the parameters, which then gave more robust estimates with error estimates. But interestingly, the underlying model that was done for that is essentially the same as the model we used in Walker Branch. This transition matrix then is, those elements are basically the same as the transfer coefficients that I showed you a few minutes ago. Now, what we were trying to do then was, of course, estimate the spiraling length. And the uptake length was fairly simple, as I described earlier. But the spiral isn't just the uptake length. Once the phosphorus is taken up, and it's in various compartments, then those are drifting downstream. And when those are associated with fine particles, those are being carried downstream quite a distance with the seston. So there's another distance that's beyond the uptake length that happens before that phosphorus actually gets back to where it can start the spiral again. And so, but we could use that model results to estimate that turnover length, that needing to know what the residence times were in various compartments and flows through those compartments and their downstream transport. But what we got then was an estimate for the turnover length, the second part of the spiral of 25 meters. Now, that was only 13% of the total spiraling length. And I think that is an underestimate. One of the results of this short estimate for the turnover length is that over the years, the uptake length and the spiraling length have been generally regarded as synonymous. So if you measure the uptake length, you basically have measured the spiraling length in the stream. And so there really haven't been any estimates of turnover length since. But as I said, I think that estimate was too low. And the work that's Pat Mulholland and Amy Rosamond have done since in Walker Branch on the seasonality of phosphorus dynamics lead me to believe that we somehow picked the worst time of the year and perhaps the worst year to measure a reasonably long turnover length. And I also think that there are longer turnover lengths in a lot of streams out there. There are two cases I'd like to show you here where there's enough information in the reports that I was able to make an approximate estimate of what they think the turnover length was. One was from the Kaparick River with 6 streams reported by Will Wolheim in 2001. And all those 6 pretty much agreed with each other. And then in the Kootenai River in Idaho and Montana. And in both cases, the turnover length was roughly equal to the uptake length. And I think these are streams that are low in nutrients, and they're also open streams, so they're autotrophic. But I think streams like that may typically have a long turnover length. And it's this long turnover length, a stream with a long turnover length, I think, where the potential for the consumers to be very effective in enhancing the spiraling by shortening that spiraling length. In other words, either by filtering particles that are moving downstream or by regenerating those nutrients in place that would otherwise be transported downstream. So that in theory, this happens if there's a long turnover length. And that theory is in this paper from 1982. And one of the things that this paper concludes that the circumstances of Walker Branch are such that the consumers in Walker Branch probably are not enhancing uptake. That is, they're not shortening the spiraling length. They may be lengthening by simple things like raising down the primary consumer compartments. But that this paper also says that where there is a strong nutrient limitation and a long turnover length, those are the conditions under which you would expect the consumers to actually enhance the uptake and shorten the spiraling length. So I think this is something that I never really pursued, but I think it's a question that deserves looking at again. Now, before I move on to or leave nutrient cycling, I need to mention a series of things that I'm not going to talk about in the interest of time, so I'll just mention them. One is the uptake of velocity. Now, the uptake length and the spiraling length have a troublesome property that they're very sensitive to stream size. So it becomes difficult to compare results from different size streams. What this uptake velocity does is scale that size effect back out and express the uptake in terms of a mass transfer coefficient or a piston velocity. So this uptake velocity has been very helpful in taking measurements of uptake length and putting them on a comparable basis among streams. Another thing that happened was that we used an isotope, and at the time, stable isotope work with N15 was not available. So there were several years there where no one could use tracers anywhere but Oak Ridge National Laboratory, where we could use radioactive isotopes. But a number of people figured out in the next couple of years that you might be able to estimate the uptake length simply by increasing the concentration of the nutrients and then adding the background nutrient, and then watching that return downstream to ambient levels. So that technique then, once it caught hold and it more or less got the stamp of approval from the Stream Solute workshop, the paper published in 1990. And so by the year 2006, when Scott Ensign and Martin Doyle published a review of these things, there'd been actually hundreds of measurements of nutrient uptake, phosphorus and nitrogen uptake, and a lot of different streams. And so that paper found a number of interesting results, one of which was that uptake velocity tends to be fairly conserved over the stream network. But the problem with this nutrient addition is that when you increase the concentration, then you tend to saturate the uptake. So you end up with an overestimate of the uptake length, which means you're underestimating the actual uptake under ambient conditions. So there have been a number of approaches to compensating for that saturation, which are coming into widespread use. And then finally, that dependence on stream size turns out to have interesting properties of its own. It turns out very roughly that the spiraling length increases almost in proportion to the stream length through the river network. And that's a property of both the network geometry plus the fact that that uptake velocity tends to remain the same or doesn't vary very much throughout the river network. And finally, then, as I've alluded to before, there was the advent of the use of N15, which happened around the year 2000, really opened up the field to studies of nutrient cycling, especially of nitrogen, but broadened the whole research field quite a bit. So that was there's been a lot of good work come out of that. And one of the major central players in that work was Pat Mulholland, who I had the good fortune of being able to work with for 3 to 4 years, where we overlapped at Oak Ridge National Laboratory before I went to the Stroud Center. So that's Pat on the right next to me, and Paul Singley, an assistant, on the left. Now, Pat just, I have to point this out. I mean, he was made such major contributions to our field, and it is a real shame that his early death took that away from us. It was a big loss to all of us. Now, I want to talk about carbon spiraling, which is a lot like the spiraling of now, carbon is a nutrient, but in shorthand, I tend to say nutrient spiraling. I'm not talking about nitrogen and phosphorus and carbon separately. But the carbon spiral, it differs from nitrogen and phosphorus in that the carbon then is destined to be mineralized and then lost to the atmosphere as carbon dioxide. So we really only have a half spiral. And we're talking about the distance from where the carbon either comes into the stream as a Lochlanus material or gets fixed by primary production and then finds its way downstream some distance before it's mineralized. And that average distance we call the turnover length, which we can calculate much like other spiraling. It's the ratio of the downstream flux to, in this case, the respiration flux expressed on a respiration per unit length basis. Now this is an average, and it's an average of widely different numbers. Because there are so many different kinds of carbon. Trees may not move very much at all. Woody debris may just stay put over its life, and whereas dissolved organic carbon, especially if it's recalcitrant, may move downstream to the ocean. So you're talking about an average of such different numbers that it's hard to get much specific meaning out of it. Also, I want to point out that this is when you're talking about a lot of different components to an average distance, that has to be weighted by each component, and the weighting is on the respiration, not on the downstream flux. So we're actually weighting it's an average of the fluxes through the system rather than an average based on the downstream flux. So what we have to do is sort of break that into components. But while we're talking about the overall uptake length, I want to relate it to the river continuum concept, which had as one of its important components this notion that downstream ecosystems are linked to upstream ecosystems because metabolically, because organic carbon is moving downstream, that leaves the upstream headwaters and is transported downstream, and then it's used downstream. So it creates this upstream to downstream linkage. And what the carbon turnover length then does is more or less put a scale on that linkage, which I'll get back to. But right now, what I want to do is mention some of these authors. The first two, Robin Van Oot and Wayne Minchell, are pictured here. on the Salmon River in 1976, working on that river continuum concept early project. I had the opportunity to work with Robin for the first few years before he retired at the Stroud Center, and I'm happy to report that he is doing well in retirement. Now, as I mentioned, Wayne, very sadly just passed away in April. Wayne, in addition to being a great benthic ecologist, he was a master at organizing field campaigns and always seemed to have several graduate students that were fun to sit with around a campfire in the evening. Then here's another shot of Wayne on the left with some of those graduate students, former graduate students, whom you may recognize. Then Bert Cushing. Bert exemplifies those qualities that the Society for Freshwater Science has of collegiality and mutual support and graciousness. I learned a lot from him as to how to interact with people. It was a real model for that. And I first met Bert when I was still at Oak Ridge. And from the time I met him, he was excited about this question: how far do particles travel in a stream? This, of course, came from his interest in the river continuum. And in fact, he was the editor of that 1983 food web paper that I described a little while ago. And it's because of questions that he asked that there's a paragraph in that paper estimating how far particles get transported in Walker Branch. But 2 to 3 years after I arrived at the Stroud Center, I got a proposal in the mail from Bert Cushing and Wayne Minchell, in which they proposed to take seston out of the stream, tag it with C14, and then reintroduce it to the stream and watch its dynamics, and among other things, determine how far it traveled before settling on the stream bed. And I said, this is a great proposal, but you really need my help. And they said, well, okay. So that was the beginning of a really productive, rewarding collaboration. And so here we are in Idaho, northern Idaho, in the Sawtooth Range. This is Smiley Creek, tributary to the Salmon River, to the upper Salmon River. And we introduced carbon-14 labeled particles to Smiley Creek. There's Wayne on the stream bank taking the picture, but as you can see, he had the camera a little out of focus. And here's Wayne again sampling the stream with the example of our seeing the slug of seston move down the stream, from which we were able to get an estimate of the transport distance of 580 meters, which translates to a deposition velocity of 0.16 millimeters per second. That's analogous to an uptake velocity. And just for perspective, a typical uptake velocity for ammonia might be 0.05 millimeters per second. So particles tend to settle to the stream at maybe a little faster than solutes get to the stream bed, but of course there's a lot of overlap. In addition to asking how far the particles go before they settle, then we want to know how long they stay on the stream bed before they take the next part of their journey down the river. And to do that involves, once the label's on the stream bed and the particles are coming back off, then to see them, you have to sieve an awful lot of water. So that's what you're seeing here. And that's going on here with Steve Thomas, who joined the project about this time and rapidly became a central and major player in this work on the seston transport. So this is watching that seston suspend off the bottom and, of course, decaying away over time as it was depleted. And from that, we estimated a residence time on the stream bed of about 5 days. So if you take a transport distance of, in this case, 750 meters, so the particle goes 750 meters, lands on the bottom, and then sits there for 5 days, and then is resuspended and moves on downstream, then it's migrating downstream at 150 meters a day, the 750 divided by 5. So that with that downstream migration velocity and an estimate for how long it might take for that carbon to mineralize, which in this case we use 255 days. I won't get into where that estimate comes from, but the product of those two numbers, the migration velocity and the turnover time, gives you a turnover length of 38 kilometers, which is a lot longer than Smiley Creek. So that means that these particles are being transported downstream significant distances where they can support the metabolism in downstream reaches and give us some sort of indication that, yes, there is significant upstream to downstream linkage through the transport of organic particles as seston down through the river system. Now, one thing we couldn't figure out is what actually was controlling the deposition velocity of particles. It didn't seem to respond. We couldn't see an effect of the things we thought would work, like water velocity and bed roughness or exchange with the hyporheic zone. But we did notice that the deposition velocity was more, for large particles, was more or less in agreement with what we might predict as gravitational settling by Stokes' law. But when the particles got smaller, then their deposition was faster than you would predict from Stokes' law. And so Steve Thomas came up with this sort of conjectural relationship suggesting that once the particles got out of the range of gravitational settling, then there was sort of a background rate at which particles made it to the stream bed. But as you can see, that was conjectural with only one point there over on the left of the small particles, and that's bacteria that Bob Hall put into a stream and measured its deposition velocity. But since then, there have been a number of other measurements which suggest that this is a good estimate of how fast small particles, or a range of estimates, I should say, how fast smaller particles get to the stream bed. But what might be, well, let's see, I got ahead of myself. Now, what about those caddisflies? Because, this was sort of the inspiration on the whole thing. Well, in Idaho, we did remove the caddisflies from Deep Creek by deoxygenating the stream, and we removed black flies from Upper Bloomington Creek with BTI. But we found very small effects. Actually, I have to admit, none of them were statistically significant. But that estimate of the removal by caddisflies, this is not to say that caddisflies aren't removing particles. The average, we calculated or estimate the average transport distance to getting caught in a caddisfly net, in this case, is about 300 meters. But the problem was that the transport to the stream bed, just getting to the bottom, was only 10 meters. The flow was really low at this time. So what's really going on here is that removal by the caddisflies is being overwhelmed just by the settling of particles onto the stream bed. With the simuliums, we got some evidence of some relatively high uptakes, but there was so much variability, we didn't see that as statistically significant. But this brings us to the question of mussels, which none of us in those days were thinking about, but there's been a lot more interest in mussels recently, and mussels filter a lot of water. And because they're endangered typically, and there's interest in restoring mussels, or because of their endangerment, there's a lot of interest in restoring mussels. And also talk about the possibility that through all this filtering, they can make the water cleaner. For example, reducing the suspended sediment concentrations. Well, I have to admit right now, I haven't done any personal work with mussels, but there are good estimates of mussel filtration out there. And we also have an estimate from Dave Strayer of the range of mussel populations in North America that might have been here prior to European colonization. And that range is 1 to 10 mussels per square meter. So if we go at the upper end of that, the 10 mussels per square meter, and multiply that times a typical filtration rate, we can get a rate at which these mussels are clearing the water of 0.006 millimeters per second, which is a piston velocity analogous to a deposition velocity. I don't call it that because they're not actually depositing. But that can be compared to this background deposition, which, as I suggested earlier from Steve's diagram, might be about 0.05 millimeters per second. So what this means is that you could have a substantial populations of mussels in the stream. They're removing particles, and they would be increasing that deposition by about 10%. And if that's the case, then what they would be doing as well, because these particles have to be being suspended just as fast as they're depositing, so that would actually decrease the suspended solids by perhaps 10 to 15%, which puts them in the same range actually as estimates of the role of caddisflies in streams. That is, they're removing a lot of particles, but more or less overwhelmed by the background rates of suspension and deposition. So we're still on this question of what controls the deposition. Early on, Ted Georgian, who worked with our project, had suggested maybe it's biofilms. So we did look at the biofilms. These are streamside flumes at the Stroud Water Research Center. And one of the things we did there, this was work with Steve Thomas, was to put rocks in the stream facing up and with a vertical face, so we would eliminate confounding by gravitational settling. Some of the rocks had been been dry and out of the stream for a year, had no biofilm, and the other ones had been incubated in the stream. What we found out was that the orientation, whether it was vertical or face up, made no difference. But whether there was a biofilm or not made a large difference. The biofilms were trapping particles, and the bare rocks were not. In another experiment that Tom Batten was involved in that looked at a number of different things, but part of that result was that we could see the deposition velocity of particles in these flumes increasing right along with the growth of the biofilm, which was a good result, except that it bothered me a little that it was correlational. We didn't have a no biofilm control. So we tried to do that I think we did that by incubating two of the flumes with biofilms and keeping the biofilms out of two of the other flumes by excluding the light and poisoning them every few days. And the result of that was that the flumes with the biofilms deposited seston about twice as fast as the flumes without the biofilm. So this suggests a strong role for biofilms in trapping particles and in controlling spiraling. And actually, I'm sort of stepping, maybe going a little too far here, but what I want to suggest is that this role that we were originally so excited about and envisioned for caddisflies as the central actors in the spiraling scenario actually might be replaced by biofilms, which are largely living. So we still have biology at work. It's just that we've moved from the caddisflies to the microbes that are in the biofilm. Now, I want to talk about dissolved organic carbon spiraling and set this up by reference to this now classic paper by Cole and his colleagues from 2007, which pointed out that you can't estimate how much carbon is being lost from the land by measuring how much gets into that the rivers are delivering to the ocean, because there's an awful lot of carbon lost along the way. And that estimate of carbon evasion as CO2 of 3/4 of a petagram per year that you see here has actually been revised upward by about a factor of 5 since this paper. So this is as bigger problem even than it was then. And one of the central questions is, where is all this carbon coming from? And there are a lot of pieces to that puzzle, but one of them is how much is coming from dissolved organic carbon, which is in itself a problem, because dissolved organic carbon, if you try to look at it through the river network, does not seem to be disappearing as fast as it should be to explain the carbon we think is coming off from the DOC respiration. This is a classic example here. This is from Jeff Ritchie in the Amazon, with the dissolved organic carbon staying virtually the same over 1,800 kilometers of travel through the Amazon basin. despite the fact that we know there is a lot of carbon dioxide coming off that river and a lot of respiration going on in the river. So to resolve this, it's been proposed by a number of people for a number of different rivers, actually, that there's a lot of input of highly labile dissolved organic carbon that comes in as lateral input or is generated from other carbon sources within the stream that is turning over rapidly and over such short distances that you really don't see it at this network level. And so this idea that there's a mix of highly labile and all the way to highly refractory or recalcitrant dissolved organic carbon, it's been around for a long time. And here's a version of that Luke Kaplan and I published back in 2003. In which we envisioned the labile forms cycling more or less over short distances and within the length of a stream order, and then less labile or intermediate forms, which I'll call semi-labile, moving downstream and, say, linking one order to another, and then a component that's recalcitrant or refractory that's basically moving through the whole river network as though it were a pipe. But the thing is, we don't know what these turnover rates are, how big these different fractions are, how far downstream we're talking about, or perhaps most of all, how important, what the contribution to metabolism is. And we could get those things if we had a tracer for natural dissolved organic carbon. But we don't have a tracer for natural dissolved organic carbon. It would be nearly impossible to come up with because it's a myriad of compounds that would have to be uniformly labeled, ranging from the highly labile to the recalcitrant and refractory. But Luke Kaplan came up with maybe what's the next best thing, which is growing trees in a C13 labeled atmosphere. So we did get a uniform label of trees, which then we put into carbon-free soil and composted and extracted that dissolved organic carbon from it and put that into the stream. This is White Plate Creek at the Stroud Water Research Center in southeastern Pennsylvania, and sampled that in the typical way. You sample a stream release over a 4-kilometer reach going downstream. And we actually did this twice. The first time, first results are published in Freshwater Biology. What I'm going to tell you about now, the second, because we're a lot more confident in those results, we did it better. But the bottom line is nearly the same in both stories. This is what we saw, the disappearance of the C13 moving downstream over 4 kilometers. And as you can see, it's not an exponential curve, which you wouldn't expect because it's so many different kinds of carbon in there, not just a single species. So, but more or less surprisingly, just by grouping these into 3 categories, we did get a good fit to that curve. So it's actually the sum of 3 exponentials, a highly labile loss, then a semi-labile loss, followed by one that statistically was not a loss at all, although there may have been a small loss there in that recalcitrant fraction. But of course, this isn't the natural DOC. This is the DOC that we got by trying to approximate the natural DOC. Now, to get from here to what the natural DOC might have done, we use these bioreactors, which are sintered glass columns through which the water flows, and with various residence times. The short residence times measure the most labile, and the long residence times measure essentially all of the bioavailable in the system. Now, by running both the C13 label and the natural DOC through these things simultaneously, we were able to get a translation of the lability profiles of the two and translate that curve, which is now grayed out at the bottom, into a virtual curve or an inferred curve of what the natural DOC tracer of the natural DOC would have looked like. And that shows us that 10% was labile, 20% semi-labile, and 70% refractory. That labile 10% had a turnover length of 100 meters, the semi-labile of 1,800 meters and the recalcitrant arbitrarily of more than 10 kilometers. What that means is that that labile is disappearing and being replaced every 100 meters along the stream, and the semi-labile every 1,800. And even sort of more surprisingly or impressively, when that's translated into uptake, that labile 10% accounts for 95% of the uptake in the stream, with the semi-labile accounting for the other 5%. And the two of them added together account for about 2/3 of the ecosystem respiration in the stream. So this supports that conjecture that the labile DOC is playing a big role. And I think it actually says that it's playing a larger role than most of us had ever thought. It gives a fairly central role to that labile DOC and allows us to sort of revise that original drawing like this, with the emphasis on the carbon, and concluding that most of the metabolism is supported by the labile DOC that originates a short distance upstream. That the reason we don't see declines through the river system is that most of that really is nearly refractory. And finally, though, that this is a little disappointing, little metabolic linkage from upstream to downstream orders, because though that semi-labile is linking those, it really contributes very little to the metabolism. So that is most of what I wanted to say, with a real quick summary saying the ratio of downstream flux to a lateral flux is a distance, sort of the basic mantra of spiraling. That the potentials for tracers to track nutrient spiraling through a food web has not been fully exploited. And I'm thinking mostly of the potential for looking at consumer regeneration in streams with long turnover lengths. That biofilms may play a role in spiraling that was originally envisioned by filter feeders. And finally, that a small fraction of the DOC does the metabolic work in a stream and does show over short distances. And I thank you very much for your time.
That is excellent, Dennis. Really, it just kind of brings back all the folks that you're able to highlight in your talk and all your collaborations. We really thank you. Obviously, your work has meant a lot, not just to the society, but to us. And to recognize, Dennis, I mean, you're a fellow of the society, so it's great to hear your words and hear your work. We do have some questions. We have some good folks out here. So just to remind you, if you send questions to Dennis on the broadcast, we're just going to send them to him. Right now, we know that Dennis is disconnected from the broadcast. We'll see if he comes back in, but if not, continue to send the questions and we'll make sure that we get them to him. Now, we do want to send some reminders. There are still activities going on today. There is some programming even now at 3 p.m. SFS Standing Committee meetings and info sessions are going on, so all are welcome to come in and visit with the committees. There's also the fun run. If you haven't seen information on the fun run, again, make sure to follow up on the emails that are being sent. And if you could head out there, do some fun running the entire week. So again, we thank you for coming up and signing up with us. It looks like we've lost Dennis' broadcast, but please continue to send the questions. I think he's back now. Okay, like Dennis is back now. Let me see what I have. Dennis, can you hear me?
Yes, I can. Can you hear me?
I can hear you, and we got some questions out here from some of the folks that were signed up. The first question comes from our good friend Jen Tank. Let me see. Jen wants to know how can spiraling theory be used to understand other particle types, for example, environmental proteins, contaminants, etc. And is there room for expanded application to these other practices? And thank you to Jen for that question.
I'd say that there definitely is room. I think you see that spiraling as a sort of a set of ideas. So I've decided over the years that it's not something that you expand and adapt to, analytically to new problems, it basically informs the way you look at the question, So where in the case of, say, environmental DNA, I think the work that you were involved in is amazing that way, that, that spiraling basically helps interpret what's going on. The idea that we have a deposition velocity, that we talk about the transport distances. It's spiraling in this case is basically just a tool to help us conceptualize and maybe express what's going on. In that sense, it's applicable widely, but I don't recommend trying to adapt the theory for specific problems.
Very good. Now, we also have a question here from our good friend Mike Paul. Mike asks, you started mentioning how we borrow concepts from cycling in lentic systems and pull on the cycling to create spiraling. He's wondering if the concept has influenced nutrient cycling in other systems, especially maybe in estuaries.
I've seen that parallels to comparing it to lake systems and even to soil. I had not familiar with anyone who talked about estuaries. I think estuaries where tidal cycles are probably are probably a dominant feature. I think estuaries might be a place where you have to rethink things. I'm not sure if spiraling actually will be of great help in talking about an estuary.
Very good. It looks like we have a question here from Ruth. She wants to know, she's curious if labile versus semi-labile is due to the proximity or the position or velocity in the water column or type of dissolved organic carbon, particularly molecule or size.
Well, there's unresolved arguments in the literature about what makes carbon refractory or labile. Different viewpoints on that. I would say we're not talking about its position, its physical position in the system. We're talking about what it is that makes it labile or refractory. And in some cases, it is from some viewpoints, it's a matter of the molecular structure. From others, it's more physical. But I would say it's not really a question of where, whether it's near the stream bottom or high in the water column or in the hyporheic zone or something like that.
Very good. Amy Roseman, she wants to know if to be able to measure nutrient uptake in a very widespread way, like in municipalities, for example, what simplified methods could you suggest, Dennis?
Could you ask the question? I missed the word. To measure it in a what way?
In a very widespread way. So, for example, in a more widespread way, like in measuring municipalities or etc., if there are any simplified methods that you could suggest.
Well, I think that I very briefly touched on using the nutrient additions, where you actually increase the concentration of the nutrient, with the proviso, with the understanding that you're not actually estimating the uptake, you're probably underestimating it. So that can be used in lots of places. Of course, I'm not actually sure whether, if you want to put anything to a river, depending on who's watching, you might need a permit to do that. So I'm afraid I don't have good answers to that question. I can say I don't have, up my sleeve, a real recommended ways to get around that. But I think there's a lot of potential for figuring out ways to measure nutrient uptake in situations like that.
We also have a question from our good friend Walter Dodds. Walter asks, do you think that your work reinforces the importance of algae in streams, even in forested streams?
Whether it reinforces it, certainly the recognition of the importance of algae has become much larger over the years since we did the original work. We we now know that algae, even in well shaded streams, is probably contributing disproportionately to the food web. And in fact, so that's one of the things that I didn't admit from that early Walker Branch work, which was that we basically assumed that it didn't matter whether the nutrient was coming from algae or from the microbes in the detritus. So we estimated that was proportional. So in a way, I would say, no, the spiraling hasn't done that, but we know that it's changed, and that influences the way we would describe the spiral, and that we would have to, that leads to some revisions of the way we see what's going on. Certainly, algae is extremely important in all ecosystems. But it occurs to me that streams with a lot of algae are the ones that are likely to have a long turnover length. And that's always been something that I would study in another life, or if I had a lot more time, would go out and start looking at autotrophic streams and looking at trying to figure out whether that long turnover length really makes a difference in the way that the consumer community modifies the nutrient cycle.
Excellent. Now, Dennis, again, I want to highlight the importance of your work. Obviously, the work that you've done for the society and what you've meant for the society. I want to just mention something that I read when you were a recipient as a fellow. You wrote, "To be kind and honest, search for the truth, immerse yourself in the system you are working in, literally where feasible. Publish your work, but don't worship the grail of publication. Balance work and personal life. Communicate to and engage with the public. Work to save our aquatic resources." I couldn't be more in awe to be able to have this conversation with you and to be able to present this award for you. So again, I thank you once again for everything you've done for the society.
Well, thank you so much, Jacob. I really appreciate the opportunity. It was a real opportunity to be able to go back and think these things through to prepare this talk. I felt like I got an awful lot out of it myself, maybe more than anyone else has.
All right, so it's the last question I'm gonna read for you. Just give you time to participate in some of the other activities that are going on today. Our last question will be from Bob Hall, and he writes, "Our scaling work of nutrient never gets close to what we might call a big river. Might we expect the scaling to hold in the Mekong or the Mississippi, some of the larger rivers that we know we've worked on?"
That's all I can say is that's a good question. I don't have an answer for that. My guess is that things start to fall apart as we scale up. The small rivers look like big rivers as long as they have some of the same characteristics. By the time you get to the Mekong and rivers like that, the big difference, of course, is that you get a phytoplankton population in the river. And a long time ago, I published a paper that talked about what might happen in a river that develops a phytoplankton population. As you move downstream and finally you have enough residence time, and the addition of new water becomes small enough so you're not just washing away the growth of the phytoplankton, then the phytoplankton can actually grow in the river. And when that happens, you should have a very long turnover length, and basically the water column starts acting like that of a lake, where you could have very strong nutrient limitation, where your phytoplankton actually draw the nutrients down to very low levels. So it's really an interesting question, but how that scaling would work, I don't have very good answers to that.
Well, thank you so much, Dennis. Again, it's been a pleasure having this conversation with you and listening to your words and listening to your work and your talk. Again, I thank everybody for showing up and for providing questions. There's a lot of great words to Dennis in the chat, and hopefully we'll see you again in some of the continuous programming that we offer throughout the week. Dennis, thank you again, and thank you everybody for tuning in. as we wave out so we could participate in some of the rest of the activities for the day and the week. Thank you all.